Hello friends, welcome to our YouTube channel, and we are studying about the topic fertilization, and this is the fourth lecture, and in this lecture we will come to know about the cortical reaction and the steps which are responsible for the stopping of the polyspermy. So let us take a look toward this figure. So before coming to the figure, let us understand what we have studied till now. The last class we have studied about how the sperm traverses through or passes through the layer of the egg, that is called as the zona pellucida, and it reaches to the cell membrane of the egg. Now after it fuses with the cell membrane, it comes through some sort of the degranulation, and that is called as the cortical degranulation. So let us understand this step with the help of the figure, and then after we will come to know about the process of the stopping of the polyspermy. So let us take a glance of this topic. So whatever here we are representing over, this is a cell membrane, or you can say the egg membrane. Okay, so this is a egg membrane, and the sperm binds with the egg membrane with the help of the juno, which is present on the surface of the villi of the egg, and the isomer, which is present on the equatorial region of the sperm. After it binds with after these two protein binds with each other, they interact, and the cell membrane of the sperm fuses with the membrane of the egg. So it can be easily seen that here is a fusion of the cell membrane of the egg along with the sperm head. Okay, and whenever it fuses with the sperm head, it now comes toward the inner part, yani cytosolic part of the egg. Before it reaches to the cytosolic part of the egg membrane, it comes to interact with the layer of the cortical vesicles, and these cortical vesicles contain various type of the enzyme, which we will learn later, understand later in the topic the polyspermy inhibition. So it come across to the membrane that is called as the cortical vesicles membrane, and what happens here, it also fuses up with the cortical vesicles, and what happens, by the time when it reaches to the cytoplasm, the cortical degranulation occur, and this degranulation results into the scattering of the cortical granule material on the surface of the egg cell membrane, and it contains various type of protein which are responsible for the stopping of the other sperm to get inside the egg membrane, and in this way the polyspermy is stopped. So let us understand the topic, and that is called as the polyspermy inhibition. So the polyspermy inhibition can be performed in various steps. So today we will be try to understand even the latest concept of the polyspermy inhibition. So let us focus ourselves on this topic. First, the first step of the polyspermy inhibition includes modification of the zona pellucida protein layer. Another one is the zinc spark. This is also a new concept, and the third one is the alteration of alteration or shading of of the juno protein. So under these three headings, we will try to understand how the polyspermy inhibition occurs in the egg. So if we concentrate ourselves on this first topic, that is called as the modification of the zona pellucida layer. So the modification of the zona pellucida occurs in the various steps. So what is that steps? Let us understand. First of all, there is a protease, and that is called as ovistatin. Ovistatin is a protease enzyme which is present in the cortical granules, and this ovistatin is responsible for cleaving of the zona pellucida 2 protein. Okay, so it cleaves the zona pellucida 2 ZP2 protein, glycoprotein. The another one is it also contain another enzyme, and that is called as the N-acetyl glucosaminidase. And this is responsible for the clipping of another carbohydrate molecule which is present or expressed on the zona pellucida membrane, and that is called as the N-acetyl glucosamine, through which the glucosyl transferase bind. The sperm glucosyl transferase bind, and this what happens, it clip of N-acetyl glucosamine in present on zona pellucida 3. So it cleaves of the N-acetyl glucosamine present on the zona pellucida, and with the help of these two protein, what happens, the zona pellucida sperm does not bind on the surface of the zona pellucida 3, and in this way, if it doesn't bind on the surface of the zona pellucida, no more sperm could bind on the surface of the zona pellucida, and it could not be able to pass through the zona pellucida membrane, and hence it could not fertilize with the ovum. So there is a barrier, and this barrier is induced by the help of these two protein, that is called as the ovistatin, and the another one is the N-acetyl glucosamine, which is present on the surface of the zona pellucida 3 protein, glycoprotein. Now let us come to understand about the another protein, and this one is the one of the most recent concept, and that is called as the transglutaminase 2. This is the transglutaminase 2. Transglutaminase 2 is present on the subcortical region of cortical. It present on the subcortical region, okay, and upon released, they cross link it zona pellucida 3 glycoprotein, and it make the surface of zona pellucida stiff and hard. Okay, so a modification of the zona pellucida membrane, zona pellucida layer, is mediated by the three type of the protease or other enzymes. The first one is oviduct, which is responsible for the cleaving of the zona pellucida 2. The second one is N-acetyl glucosaminidase, which is responsible for the clipping of the N-acetyl glucosamine, which is present on the layer on the zona pellucida, through which glucosyl transferase of the sperm binds. Okay, after this modification of the zona pellucida 2 and the 3, the no more sperm will be able to bind with the zona pellucida membrane, and hence it could not traverse through the zona pellucida membrane. And most recently, another one is the Transglutaminase 2, which is present in the subcortical region, and whenever it is released upon the sperm reaches to the cell of the ovum, it is get released and it binds with the zona pellucida 3, and it cross-linked zona pellucida 2, zona pellucida 3, and after it cross-linking, it make a strong and hard barrier on the zona pellucida 3, and so that the normal sperm could bind, and in this way the first type of the boundary which is created by all these cortical granules. Okay, and in this way the polyspermy could be stopped. The second one is the zinc spark. This is also a 4 or 5 year concept. You know what happens? The second one is the zinc spark. After the sperm reaches egg membrane, after the sperm reaches inside the egg membrane, okay, a impulse, a spark of the zinc occurs, and this spark of the zinc occurs in millions, means the millions of the zinc comes out, millions of the zinc spark comes out on the surface of comes out of the surface of the cell membrane. First, what is the benefit of the zinc spark? In the clinical diagnostic, it is seen that whenever the egg fuses with the sperm, it creates a zinc spark, and the intensity of zinc spark says or defines the how vigorously the cell combines, how the successfully the egg combines with the sperm. And the second is that whenever the zinc sparks occurs, means once time the zinc comes out from the egg cell membrane, and it comes to on the surface of the membrane in millions in number. And what does it do? Basically it just inactivates, or it kills, or you can say it deactivates some of the proteins like the acrosine and MMP2s are the protein and responsible for the sperm to just traverse through the cumulus and the zona pellucida membrane. So it binds with this acrosine and MMP2 and it inactivates all these type of the protein. Beside this, it also responsible for inactivation of all those proteins most of the proteins which are involved in the capacitation of the sperm. So this one is the most important phenomenon where it makes the shield of the zinc, means it make a zinc shield on the cumulus and as well as on the surface, or you can say the zona pellucida layer. Okay, and in this way the polyspermy could be easily stopped. So what does it do? It the spark contain millions of the zinc. Okay, millions of the zinc. Experimentally, the zinc spark could be seen with the flash of the light after fertilization. Okay, contains millions of zinc and it inhibits acrosin MP2 protein and other protein utilized for capacitation. And third, it makes a zinc shield on cumulus so that no more sperm could bind. So this is the importance of the zinc spark, which often occurs when the egg and the sperm binds together. Now the third one is the shedding of the juno protein. Third one is shedding of after sperm enters into the egg membrane, all the juno proteins is released from the egg surface, and so that no more juno is available for isomo to bind with. So this is another layer, interaction of zumo with isomo. Okay, after the one of the sperm enters the cell membrane on the egg membrane, then what happens? All the juno which is present on the surface of the ovum get shed off or get released, and after that, whenever even though the zona pellucida, even though the some of the sperm which passes through the zona pellucida membrane, so its isomo is could not bind with the juno protein because it has got released from the egg membrane, and in this way the sperm could not bind and fertilize with the ovum, and in this way the polyspermy could be stopped. So we have come across the three reasons or the three barriers the nature has provided to the cell so that it could not allow the polyspermy to occur. Now let us understand what after this event. Right now let us take an account of the egg activation and the fusion. Now egg activation and fusion. Okay, right now the sperm reach to the cytoplasm of the egg, and what happens thereafter? Thereafter, actually, there is a surge of the calcium ion, and this calcium mediated the MAP kinase inactivation occurs, and this MAP kinase inactivation will lead to the degradation of the cyclin B and the saccharine. Okay, what happens? What happens after it reaches? After the sperm reaches, after sperm reaches to the egg, okay, what happens? It inactivates MAP kinase, and this will result into the cleavage of cyclin and the saccharine. Okay, what happens right now? The ovum is in the state of the metaphasic arrest of the meiosis 2. Okay, whenever the sperm enters in the cytoplasm of the egg, then what happens? The surge of calcium ion occurs. The surge of the calcium ion occurs, and this calcium ion reaches to the egg nucleus. Okay, after it reaches to the egg nucleus, what happens? Okay, with the multiple reaction, not egg nucleus, in the cytoplasm only, so it undergoes the multiple reaction. Okay, will not go through the details what exactly the reaction was. So after the surge of the calcium ion, what happens? It inactivates the MAP kinase, which results into the degradation of the some cyclin, and this degradation will lead to the activation of the egg. This will lead to the activation of the egg and to proceed toward the other step of the meiosis, that is metaphase, anaphase, telophase. Okay, so it undergoes the other part of the meiosis 2 process, and the saccharine, which was responsible for binding of the chromosome on the equatorial plate, get also degraded, and right now here this led it to move the chromosome toward its other poles, and in this way the cell of the egg, or you can say the ovum, completes its meiotic steps. And then after, what happens? Now the fusion of the male sperm pronuclei and the female egg pronuclei occurs, and in this way the fertilization gets completed. After it, the fusion of the gametes, the zygote is formed, and this zygotes undergo further division and division to form an embryo. So this was the this was all the steps which we come through in the fertilization process. Hope you enjoyed the session. If you like it, if you enjoyed the session, please don't forget to like, share and subscribe. Thank you. Thanks for the watching. We will meet in another lecture. Thanks.